red and blue. Yeah. So I'm hearing codominance. Is that what you were going to say? Okay. Let me open up this chat here so I can see what my friends at home are saying. So what do we think this is? Virtual students, you can put it in the chat. Awesome. Co. So remember, co means together. So we have a homozygous red, we have a homozygous blue, and when we cross them, the heterozygous is showing both together. Remember, co means together. So we see the word and when we describe this phenotype, blue and red. All right, how about this one? This one? So these are horses. We have a brown horse and we cross it with a white horse. And here's its baby, the foal, who got a chicken wing. He's very excited. So which one is this? Codominance. So if we looked closer at this horse, we would see both white hairs and brown hairs, both together, and brown and white. All right, how about this example? So we're crossing a red or a brown horse with a white horse, and their babies are cream colored, also known as palomino. Yeah. It's not codominance on this one. So this is an example of incomplete, right? Oops, sorry, let me go back. So the heterozygous is not brown, like it's one parent. It's not white, like it's one parent. It's a third blended color of the two in the heterozygous. So this is called incomplete. Okay, neither white nor brown was dominant, and this is an incomplete dominant example. All right, I know this is not how you make a panda bear, but this example was just too darn cute not to share. So what would this be showing an example of? This would be fun if this is how you made a panda bear, if you crossed a polar bear and a black bear. So this example is trying to show if we crossed a white and a black bear and all of the babies have both white fur and black fur. So this would be co again, and. When we use the word and, we're talking about codominance. All right, how about this one? Incomplete, yes. So again, remember yesterday I talked about mixing colors in art class. So oftentimes we see the heterozygous and an incomplete dominance as a blending of the two, like a third. Like if we took black and white paint and we mixed it together, we would get gray paint. So this is the example of an incomplete dominance. So the horse is not black and white, it's a third totally different color, gray. All right, so moving us on to today's topic, we're gonna do a warm-up, genetics day 7. Then we'll take some notes and take a look at 2 unique types of inheritance patterns where we're looking at multiple alleles, meaning more than 2, and X-linked traits. So traits that lie on the X chromosome, the sex chromosome. There will be a homework assignment due tomorrow. There'll be time at the end of class to work on that. And so our essential question today is, how do you set up a Punnett square using superscripts? So we'll talk quickly about what a superscript is. As in multiple alleles, and blood type is going to be the example we use for multiple alleles, and sex-linked traits. So hopefully after you do your homework tonight, you're going to be able to say, Ms. Klepper, I can set up and determine crosses using superscripts on alleles for multiple alleles like blood type and sex-linked traits. All right, so today, when we get into day 7 notes, you are going to be filling in Punnett squares where you must include a superscript along with the chromosome. So what is a superscript? Well, super means above. So think about like Superman. He flies above us, right? Super. So superscripts, we write them up in the upper right-hand corner like this, okay? You see these in math when you're squaring something or tripling something in algebra, right? Superscript. A subscript is when you write it at the bottom. We're not going to do any subscripts. You see this a lot in chemistry, okay? When you write oxygen, you write O2, and the 2 is a subscript. You write it down at the bottom. So today we're going to be doing Punnett squares where you must use a superscript, okay? All right, so let's open up your notes to day 7. And I don't know how this happened, but I messed up the dates on my notes, on your notes. So today's not Wednesday, it's Thursday, April 15th. We're already halfway through this month, which is crazy to me. May probably will go even faster. All right, so in our warm up here, we're just practicing the 2 things we just went over, codominance and incomplete dominance. So this first example here is a codominance inheritance pattern. When we cross a red flower, big R with a white flower, all of the offspring are a phenotype that has both red and white petals. So I want you over here to cross 2 heterozygous flowers on this Punnett square. So you're crossing 2 of these big R, little r. So 2 heterozygous flowers. And this should be fairly simple. You guys have been doing this for over a week now. So hopefully this is not anything that's too challenging for you. All right. So you should see the 3 different genotypes in the Punnett square in the offspring. So what is the genotype ratio of this cross? So somebody in my class is saying 1 to 2 to 1. They are correct. So the first number is how many are big R, big R. The second number is how many are heterozygous. And the third number is always how many are homozygous recessive. So again, that's from back on day 3 when we did Punnett squares and genotype ratios. All right, so the phenotype ratio here is a little bit different than what we did on that day 3 because we have a third phenotype, right? So this flower that's red and white is not, it's not red and it's not just white, it's red and white. So what do you think the phenotype ratio of this cross would be? So 1 to 2 to 1 again, right? So we have 1 red right here. We have 2 that are going to be red and white. And those are right here, our heterozygous ones. And the last number in our ratio is the number that are white, 1 white. So in this case, the phenotype and the genotype ratio are the same, 1 to 2 to 1. All right, so now let's look at an example of incomplete dominance. So this is different because we don't have red and white in the heterozygous phenotype. We have a pink flower. So you have 2 crosses over here. I want you to fill in your Punnett square and then answer the questions about them. So try it out yourself. All right, hopefully filling in the Punnett squares is easy breezy for everybody. Answering questions sometimes can get a little tricky, so I'm just gonna remind you, always read very carefully what the question is asking you. That's just good test taking skill to learn. So the number 5 is asking, what % of the offspring will have a heterozygous genotype? So what did you guys write? Somebody just whispered it in my class. Was that you, David? What did you say? 100%. You are correct. So all of the offspring are big R, little r. That's what heterozygous means. So if you're like, I don't know what heterozygous means, you got to go back to day 1 and day 2 and look at vocab, because maybe you need some practice with that. All right, what % of the offspring will have white flowers? 0%. Remember, to have a white flower, you needed to be little r, and none of our offspring are little r, little r. They're all 100% heterozygous and pink. All right, so for the next cross, we crossed a red and a pink flower. So what % will have red flowers? What did you guys write? 50%, right? Half. All right, and I think number 8 is the hardest question on here. So it's asking, what will be the ratio of red to pink flowers? It's not asking about white. It's not asking for the genotype or the phenotype ratio. It's just asking, what is the ratio of red to pink? Yeah. Yes, 2 to 2. Awesome. 2 to 2. So there's 2 red to 2 pink. So that's how you do ratios. I know we've been learning the order and everything, but this question was asking just for how many red to how many pink. All right, let's move on to today's topic. And I got a blood splattering for you here because we're going to be talking about blood. All right, so we're going to talk about blood because blood type is unique. And we say that their blood type has what we call multiple alleles, meaning that there are more than just 2 alleles out in the world. Okay, there are actually 3 possible alleles that humans can have for blood type instead of 2. So there's 2 dominant alleles. You can have a dominant A, you can have a dominant B. But you also can have a recessive allele, which we use a lowercase I. So the I represents whether it's dominant or recessive. And the A and B end up being superscripts on just the dominant alleles. Okay, only the dominant alleles get the superscript. The little I has no superscript. Yeah. So AB is rare on its own, and then negative has to do with whether there's a protein on the outside of the blood type. So it must just not be very common in the population. But why it's so rare? AB blood in itself is rare. Sweet. All right, so I want to remind you that you still only get 2 alleles for blood type. Okay, you get one from your mom and one from your dad. That doesn't change. What changes here today is that there are 3 possible alleles out there in the human population instead of just 2. So there's 2 dominant alleles and 1 recessive. And because of this multiple alleles, blood type is unique. Okay. When you have type A and type B blood alleles together, they actually end up being co-dominant. And this has to do with antibodies on the in the plasma and antigens on the blood cells. So I'm going to give you just a really quick explanation of this. If you are interested in learning more, there's a link here you can go to. So you probably know that there's 4 blood types, at least the groups. Then there's also positive and negative, which has to do with another protein that you find on the outside, but I'm just going to talk about the 4 types of blood. So if you have type A blood, on the outside, you have these little markers that we call antigens for A. And this is how your body knows that this is your blood cell. This is the right type of blood for your body. In the plasma of your blood, which is the liquid of your blood, we're going to learn about this, one of the last things we do in this class. In the plasma of your blood, you have antibodies for type B blood. Okay, see how these are shaped? And that means if type A people, blood type A people get B blood, your body is going to see it as foreign and it's going to attack it. Your immune system is going to attack B blood because it's seeing it as foreign, and this is not good. If you need blood and you're getting a donation and your body starts to attack and kill those blood cells and you need them to survive, that's very, very bad. The opposite is true for type B blood. They have B antigens on their cells and they carry the antibodies to kill A. Okay, they're antibodies for A. People who are type AB blood have both antigens. This is why we call AB blood co-dominant, because both A and B antigens are on these cells. And these people don't carry any antibodies in their plasma, which means they can accept blood from any of the other types because their body's not going to attack it. And then lastly, we have type O blood. So type O blood has no antigens on the outside of their blood cells, which makes them great donors because other bodies won't attack them because they won't recognize them as foreign. But type O, right, type O blood will attack because they have antibodies for A and B. So they will kill type A and type B blood cells if they're introduced. So no antigens on type O, but they carry the antibodies for both. So yes, type O blood can only accept type O blood. All right, so that's a little very, very quick explanation. There's way more involvement when we start talking about positive and negative blood types, but we're just going to focus on the 4 groups of blood today for our genetics unit. If you want more information, there's a link here you can click on if you want to learn more. All right, so here are those alleles again that I wrote out. So you can have a dominant A allele, you can have a dominant B allele, or the recessive allele, which doesn't have a superscript. So again, little I is recessive, just like it has been. When we write a recessive allele, we use a lowercase letter, and the uppercase is dominant. And when you have A and B together, they're codominant. So let's take a look at the different genotypes that you can have for each of the blood. So if somebody is a type A blood, there's actually two possible genotypes they might have. They can be homozygous dominant for A. They can be heterozygous for A, where they have a dominant allele and a recessive allele. Same thing goes for type B blood. You can be homozygous dominant for B, or you can be heterozygous for B. This is still only two alleles. It's just we're using a superscript now. So don't get confused that this looks like it's more than one allele or more than two. It's just two. Type AB blood has one of each dominant allele, so a dominant A and a dominant B. This is still considered heterozygous, by the way, because you have an A and a B. Those are two different alleles. So this is considered heterozygous because of that. And then type O, this is my genotype right here, I'm type O, has two recessive alleles, little I, little I. By the way, my dad was type A blood, and so I know my dad's genotype had to be heterozygous because I'm type O. So my dad had to give me his recessive allele to make me have type O blood. I don't know what my mom's blood type is, but she would also have to be either heterozygous A or B or type O as well. All right, so let's try, let's fill in a Punnett square using our new skill of using superscripts. So we're gonna cross somebody that's type A with type B. And so I wanna remind you here that when sperm and egg form, these two alleles are still gonna separate. They're just taking their superscripts with them this time. So this person can pass on a dominant A or a dominant A. Since they're homozygous, they're only going to pass on one type of allele. And then this person being heterozygous can pass on dominant B or the recessive I. So when we set up our Punnett square now, we're just needing to write the dominant I with the superscript for the alleles, for the gametes of these parents. All right, so just like every time we filled in a Punnett square, there's some rules that you're gonna have to follow here. So one rule is that A always goes before B. Okay, so if the baby's gonna have an A and a B, A always goes before B. Just like in the alphabet, and we call it type AB blood, we don't call it type BA blood. And then if there's a dominant and a recessive, you still write the dominant first. Okay, that has not changed. And you always keep the superscript with the dominant I. Okay, so keep it with it. It gets a little tighter in the Punnett square, but you can do it. All right, so now we can answer some questions once we filled in the Punnett square. What % of these offspring are going to have type A? So these two will be type A, right? By the way, you don't have to memorize the genotypes. I mean, it's really not that hard. It's either they're homozygous or heterozygous for A and B. AB and O blood have the same genotype, but I'll always give this to you on a test. So I will give this to you. So you'll have it to reference. So yes, 50% are going to have type A blood. And then what percent will have type B blood? I'm sorry, AB blood. The other 50%. Riddle me this. Can these parents have a type B blood baby? I mean, one of the parents was type B. They cannot. Because the type A parent was homozygous for A, they can't have a type B blood baby. They can't have a type O baby either, by the way. All right, let's do one more. So I want to see you fill this one in yourself. I want you guys to try it yourself first before I do it. This is how you know if you're doing it right, is do it yourself. And then check your work. And remember your rules, dominant goes before recessive. All right, so number 5 asks us, what percent of the offspring will have type A blood? So what do we got? These 2 out of 4, right? They're going to be A, so that's 50%. And then number 6 asks, what percent of the offspring will have a heterozygous genotype of any kind? 100%. Right? All of the offspring are heterozygous. They all have one dominant allele and one recessive allele. This is where blood type is just fascinating because these 2 parents, one of them has AB blood, one of them has type O blood. They could have a type A or a type B baby. They wouldn't be able to, the parents and the children would have different, potentially all have different blood types because of how A, B, and O work. All right, so you will have a page of blood type Punnett squares that you have to do for your homework today, but let's move on to X-linked traits. So our second topic of today. So there's another type of inheritance that falls on the X chromosome only, okay? Remember, an X chromosome is a sex chromosome. We talked about this last Friday when you were flipping to see what the sex of your baby would be. This is the one, the 23rd pair of chromosomes that determines the sex of your offspring. And we have a couple examples of things that lie on the X only. Okay, so one example of this is color blindness. So not being able to see colors the same way as a normal sighted person, and hemophilia. Hemophilia is far more serious than color blindness. People can live with color blindness. Hemophilia is a disease that doesn't allow you to clot your blood as well. And so people can bleed to death. And people did bleed to death, especially back in the day when medicine wasn't as developed as it is now. So let's start by talking about color blindness here. So this is a color blindness test. People with red-green color blindness cannot see the number that's inside of this circle. It's mainly going to happen with males because of it being X-linked, and I'll talk about that just in a moment. Females can be colorblind, but it's much more rare because of this being an X-linked trait. If this is the first time you're seeing this and you're finding that you're colorblind, it's okay. It's not the end of the world. People live with color blindness. It gets a little tricky when you're learning how to drive a car because of red and green stoplights. You have to actually learn where the light is on the order because red and green is oftentimes switched or looks different to you than it does to others. And then hemophilia is the bleeding disorder where you don't clot your blood properly. So you could have prolonged bleeding, nosebleeds could happen more commonly. And there could be internal bleeding that can cause death with hemophilia. It's much more serious, right? Not being able to clot your blood is a serious problem. All right, so what is an X-linked trait? So it's a trait that's located on the X chromosome. Because males only have one X, they can inherit a disorder on the one X. Okay, they only get one chance to get the right allele. They can get, if they get one of the alleles that causes the disorder, they're going to have it. Females, on the other hand, because we're XX, so males are XY, right? And females are XX. Because of this, females have to have both of their X's have the disorder in order to have it. So it's more uncommon in females to have an X-linked trait because, first of all, these traits are rare on their own. And then it's rare to have a female that has both her X's affected. She has to have a father who has the disorder because he has to give his affected X to his daughters. And then his mom either has to have it or be a carrier. She has to be heterozygous for it. All right. So there's a lot of writing here. I'm not going to go through all of this because I kind of am talking through it. The big thing to note is that there isn't anything on the Y chromosome. Okay, these traits are found on the X because the Y is shorter and these traits just don't lie on the Y chromosome. And all the examples we're going to focus on are X-linked recessive, meaning that the disorder lies in the recessive allele. Okay. So and another thing to note here is we have to write a superscript and we must include an X and a Y when we're doing these. So only the X is going to get the superscript because we need to know the sex of the child as well as whether or not they have the disorder. Okay. All right, so let's first just fill in a Punnett square without any alleles and just remind ourselves how it is that you have a male or a female baby. Okay, so I've got dad up here. He's going to give either X or Y. And I have mom over here on the side. So just fill in the Punnett square with their letters to see how you get a male and a female baby. So I want you to circle the female offspring. So circle the ones that are going to be female and put a box around the ones that are going to be males. So XX is a female, XY is a male. There's a reason I'm having you use circles and boxes, and we will talk about that more tomorrow. All right, so who determines if the offspring is going to be male? Dad, right? Because if he gives his X, he's going to have a daughter. And if he gives his Y, he's going to have a son. Okay, this is important to understand when we start looking at pedigrees tomorrow, because fathers always give their sons a Y. So fathers can't give their sons an X-linked trait. And what is the only sex chromosome that mom can give to her offspring? Is X. And dad can give X or Y. Okay, so just understand that understanding that is important to understand how these traits are passed on. And when we start looking at Punnett squares, pedigrees tomorrow and Tuesday. All right, so we're going to look at an example here. We're going to look at color blindness, the inability to see colors correctly. And this is an X-linked recessive trait. And so here are the genotypes that can occur. Okay, we're going to use a B to represent color blindness. Because it's recessive, the recessive allele causes the color blindness. Okay, so males are either X big B, dominant, they have the dominant allele and they have normal vision, or they can be X little b, Y, and then they are colorblind. So all the males in class right now should know what their genotype is. Maybe you're not sure if you're colorblind or not, but if you know for sure that you're colorblind, then you would know your genotype. And then males cannot be carriers. So what a carrier is it's a person who carries the allele for the disorder, but they actually don't have it. So males can't be carriers. They either have it or they don't have it. Females, on the other hand, have 3 possible genotypes. Because we have 2 X's, we can be homozygous dominant and be normal vision. We can be heterozygous, where we have normal vision, but here's that carrier where we're carrying the little b, and we could pass that on to our sons or our daughters later in life. And for a female to be colorblind, she has to be homozygous recessive. Both her X's have the little b. All right. So let's cross a normal visioned male with a female carrier. So a normal male is got, so here, I already wrote your X's and Y's, so you didn't forget to put them on there. Normal vision male has the dominant allele, and mom is a carrier. So she's got 1 big B and 1 little b. Now, rules again, right? We have rules when we fill in Punnett squares. Dominant still goes before recessive if there are both, and then X always goes before Y. Okay, so don't ever write the Y first. All right. So just like any other time we've done Punnett squares, once we have it filled in, we can answer some questions about it. So my first question to you is, what are the % chance that their sons only will be colorblind? What do I mean by sons only? So what I mean by that is if the doctor says, congratulations, you have a boy, what are the % chances that that son will have color blindness? 50%, right? This one right here is going to be colorblind. So of their sons only, 50% will be colorblind. How about their daughters? If we look at just their daughters, 0%. Neither of the two daughters are going to be little b, but what is their chance of a daughter being a carrier? 50%. So if they have a daughter, she has a 50% chance of having being a carrier. All right, let's do one more of these and then I'm going to send you on your merry way to do your homework. So let's cross a colorblind female. So we got to remember what that means. And I know you can look above to see if you can't remember what that looks like. With a normal visioned male. So normal means he has the dominant B and his Y. So fill in your Punnett square. Normal vision dad, mom has color blindness. And you should see, because mom is colorblind, that her sons. Yep. If a female has an X-linked trait, then she's going to give it to all of her sons. Yep. Yep. If your dad has it, females are guaranteed to be carriers. Absolutely. Yep. Awesome. You guys are starting to understand how this works. That's good. All right. What are the percent chances of them having a child with a child? So either sex with color blindness. 50%. Right? It's the same chance of having a male is 50%. What percent of their sons only will be colorblind? 100%. So if the doctor says, congratulations, it's a boy, he's colorblind because mom was colorblind. And then what percent of daughters will be carriers? That's also 100%. Because remember, if mom has got color blindness, she has to give her little b to her daughters. And if dad was normal visioned, then he gives his big B. So 100% of the daughters will be carriers. All right. So for your homework, you are going to be first looking at X-linked traits. Another example of an X-linked trait is eye color and fruit flies. I know, you're like, why do I care about fruit flies? Scientists actually have historically studied fruit flies for genetic reasons for many, many, many years. And they found that eye color in fruit flies is X-linked. So red eyes are dominant over white eyes. So the first thing you're going to do here is you're going to write what are the sexes and the eye colors of these flies. So I want, what is the sex? So you got to look at the X and the Y, and then using big R is red and dominant over white, you're going to write what are their eye colors. For number 2, you're going to do the opposite. They're giving you the sex and the eye color, and you have to write the genotype. Then you're going to cross 2 fruit flies for number 3 and 4. And in the next page, you're going to do a hemophilia example. And then on the 3rd page, you're going to do some blood type questions. And I just want to say, make sure you're paying attention to what the questions are asking. Okay, that's a good test taking skill to learn. So make sure you're reading everything really carefully. All right.